Okay, now my microphone is working. So we are going to be talking about lipids today, or that's where we're starting. We have a couple of different types of lipids. Our first type is triglycerides, and triglycerides are used for long-term energy storage. And we talked about that a little bit last time. This long-term energy storage, another word for it, is fat tissue or adipose tissue. In humans, when we consume extra calories and extra carbohydrates, what our body does is if we have too much, if we consume more calories than we need to sustain us for the next couple of hours, our body takes those extra calories and extra carbohydrates and turns them into triglycerides. And triglycerides are really efficient energy storage. And so we use them for long-term energy storage. I kind of think about, for some reason this is the analogy that works for me, is I think about adipose tissue, like who buys, or I guess most of you may or may not buy your own groceries, but at home, how many of you buy your paper towels and your toilet paper at like Sam's Club or Costco? You get the big, huge packs. Well, you get these big, huge packs of toilet paper. You don't want that sitting in your bathroom, right? So what you do is you take a couple of packs out, you put it maybe under your sink, you store it around in a basket or whatever, and then you put the rest in the garage. That's your long-term storage for all the extra stuff we have. That's kind of what adipose tissue is in your body. It's the extra rolls of toilet paper that you're not going to need for another couple of months. So your body kind of shoves it in deep, efficient storage in the garage for later use. And that is what triglycerides are. Now, triglyceride, again, looking at the name, that prefix tri, what does tri mean? 3. Tri always means 3, like a tricycle, has 3 wheels. So a triglyceride is going to be 3 fatty acids. We're going to talk about what that is in just a second. And 1 glycerol molecule. So triglyceride is 3 fatty acids plus a glycerol. Okay, so what a fatty acid chain is this down here. It's a whole lot of carbons and hydrogens with one little non-carbon hydrogen group at the end. A couple of oxygens at the end, but pretty much everything else is carbon and hydrogen. There are these big, long chains of carbon and hydrogen. And a glycerol is this molecule right here. And notice that glycerol, you don't have to memorize the structure necessarily. You do have to know that a triglyceride is made of 3 fatty acids and 1 glycerol. But that's all you really need to know about the structure. But this glycerol has 3 OH groups, and that is where those fatty acids are going to bind. So 1 fatty acid is going to bind there, a second one will bind there, and a third one will bind there. Okay, so we have 3 fatty acids down here. They'll each bind to 1 spot on that glycerol molecule. Okay, so this is what a triglyceride will look like. This is the glycerol up here. Sorry, my pen is too thick. The glycerol down there, and then these are the fatty acids. So we talked with carbohydrates about the monomer and the polymer. Remember, the monomer in a carbohydrate is a monosaccharide. The polymer is a polysaccharide or a complex carbohydrate. Those names were nice and easy. It has mono and poly right in the name. It was really easy. Unfortunately, for triglycerides, that's not as true. So in fatty acids or triglycerides, the monomer is the fatty acid chain. Okay, so the monomer is going to be the fatty acid chain, and the polymer or the macromolecule those words kind of mean the same thing, is the triglyceride. Now we got to talk about a couple of characteristics of fatty acids and triglycerides in particular that make them distinct from other molecules. The first one is that fatty acids are what we call hydrophobic. And I bet you guys can figure out what this word means. What does that suffix phobic mean? Scared of or afraid of, right? Like a phobia is something you're afraid of, scared of. What does hydro mean? Water. So hydrophobic means you're afraid of water. So fatty acids, they're not really afraid of water, right? They're molecules, but they repel water. And this should make sense to anybody who has ever tried to clean peanut butter off of a spoon, okay? Anytime fat comes in contact with water, it repels it, right? If you try to rinse off peanut butter from a spoon, it's not going anywhere. You have to use soap and you got to scrub it off. This is also why if you mix oil and vinegar, even if you shake them up and mix them really well, eventually they're going to separate right back out because oil and water, fats and water, do not mix. Fats are hydrophobic. Okay, so we're going to talk more about that in just a second. That's going to come back around. But fats, triglycerides in particular, are hydrophobic. They repel water. Now, the other thing we need to talk about is the difference between a saturated fatty acid and an unsaturated fatty acid. And those are probably words that you have heard before when talking about food, right? We talk about saturated fatty acids or saturated fats and unsaturated fats. But a lot of people may or may not know what that means. And with fatty acid chains, there is a difference in structure when you have all single bonds versus when you have all double bonds. So what I mean by single bonds is these are carbon molecules and hydrogen molecules. This is representative of what these molecules look like within the body. And those lines in between the molecules, so we have C line attached to the H. These lines are bonds. Okay. And when there is one line, that is what we call a single bond. When there's two lines, that's a double bond. Okay? Now, all you really have to understand about this, there's a lot of complicated chemistry here. I'm trying to kind of surface level it as much as possible. So try not to think too hard about it and just kind of take what I say at surface level. When a molecule forms single bonds like this, it is capable for them to bond in a straight line. Okay, they'll just form a straight line, just like we have it represented here. They'll line up together in a really nice straight line. However, when you add double bonds, like you have there, a double bond is going to add a kink in the chain. It stops the chain from being straight. Now we call the one on the left, this is a saturated fatty acid. This one right here. So a saturated fatty acid has all single bonds. And an unsaturated one has some double bonds. And you can see that in the picture over here, that this saturated fatty acid has all single bonds in that fatty acid chain between those carbons and hydrogens. And the unsaturated one has some double bonds. Now, the way that you can remember that, if this helps, is saturated, that word saturated means something is full, right? Like if a sponge is saturated with water, it's holding as much water as it possibly could hold. It is saturated. With a saturated fatty acid, it is called that because if you look at all of those carbons, they are holding as many hydrogens as possible. They each have 2 hydrogens attached to them, right? If you look down the chain, each and every carbon has 2 hydrogens. The one at the bottom has 3. Okay, so it has as many hydrogens as it possibly could have. However, when you look at this double bond, notice that those carbons do not have as much hydrogen as they possibly could have. Each of those carbons only has one hydrogen. So a saturated fatty acid is one that is saturated with hydrogens. An unsaturated fatty acid is one that does not saturated with hydrogens. Okay? It has fewer hydrogens than it could possibly have. And that has to do with the fact that this is not something that you really need to know, but carbon is a molecule, is capable of forming 4 bonds. 4 and only 4. So if you count the bonds on each of those carbons, this has 1, 2, 3, 4. 1, 2, 3, 4. Right? Each of those carbons is forming 4 bonds. When you add a double bond in here, that takes away a spot for a hydrogen to bond. So we have less hydrogens. So again, a saturated fatty acid is one that has all single bonds and is saturated with hydrogens. An unsaturated fatty acid is one that has double bonds, which causes kinks in the chain, and it makes it so it is not saturated with hydrogens. Now, why that is important when it comes to food and energy storage and things like that is saturated fatty acids. So these are a storage form. Again, what our body is going to do is they're going to take these triglycerides and they are going to store them in the garage until we need them. And so they line them up as efficiently as possible to take up as little space as possible. Okay, so I want you to think about if you're trying to store something, you have some shelving units in your garage, okay? And you're trying to store as much as possible of something. Are you going to be able to make neater stacks with something that's a square, like in a straight line? I can't think of anything. Like you're trying to stack up bricks or something. Something that is square and has square ends. Or are you going to be able to store more stuff if you're trying to store beanbag chairs? Okay, think something that is irregular. Okay, those saturated fatty acids, because they line up so well, saturated fatty acids can form these straight lines. And they will line up next to each other and form this really, really compact and efficient substance. Okay? Unsaturated fatty acids, if I was to draw, within this same, trying to fit as many fatty acids in this area as possible. If they are in straight lines, I can pack in tons of them because they'll stack right on top of each other and I can pack them in. But with unsaturated fatty acids, if I was trying to put put as many as possible in that same area, but they all have big L's in them, right? I'm not going to be able to fit as many because the structure is physically bent. They take up more space. So what this means for fats made out of saturated versus unsaturated fatty acids is those saturated fatty acids make a more compact and efficient structure than the unsaturated ones. So when we're talking about food, saturated fatty acids are going to be the ones that are solid at room temperature because of that really, really compact and efficient structure. So can somebody think of a food product that is a fat that is solid at room temperature? Butter is a great example. Peanut butter is another one. Peanut butter is solid at room temperature, but butter is a really, really great example of a saturated fatty acid because butter is a solid, unless you heat it up. But at room temperature, it is going to be a solid. So on that same line, can you think of something that would be an unsaturated fatty acid? Not solid at room temperature? Vegetable oil, olive oil, avocado oil, coconut oil sometimes. But olive oil or vegetable oil is a great example. Avocado oil too. There's lots of examples. But unsaturated fatty acids are a liquid at room temperature. Okay? So those are our triglycerides and our lipids, or our first type of lipid. That's a triglyceride. Make sure I said everything I needed to. All right. So our second group of lipids are called phospholipids. And phospholipids, their main purpose, so again, our triglycerides, their main purpose is this one right here, this long-term energy storage. That's the main purpose that they serve within all types of cells. Phospholipids, their main purpose is that they create membranes. So every single cell in every single organism that exists on this planet or any other planet has a membrane. We're going to talk more about specifics of membranes in the next chapter. But a membrane is just a layer that separates the inside of a cell from the outside of the cell. Okay, it's the thing that makes a cell, the outside of the cell. And all of those membranes, their main component are phospholipids. Okay. Now, the structure of a phospholipid is very similar to a triglyceride with one major difference. So a phospholipid has a glycerol, just like we had before, that same glycerol molecule. But that glycerol molecule, remember, it has 3 spots to bind something. In a triglyceride, we bind 3 fatty acids. But in a phospholipid, we bind 2 fatty acids. And with that extra spot, we add a phosphate group. Now, the reason that that is important is this phosphate group right here has a charge on it, that negative charge. And that negative charge does something really important to this molecule. It makes this part of the molecule, and only that part of the molecule, I don't know why I can't spell that word today, hydrophilic. Okay, so we talked about before, what does hydro mean? Water, and philic is the opposite of phobic. It means loving or attracted to. Okay, so adding that phosphate group, that little charged piece, to this molecule makes a piece of that molecule hydrophilic. Okay, meaning that it is water loving. Another word for that is polar. Those all mean the same thing. Polar, hydrophilic, water loving. That means that that part of the molecule can and does interact with water without being repelled away. All right, but in a phospholipid, the bottom part, these are still the fatty acids. These are still hydrophobic. Okay, so non-polar is hydrophobic. So what that means is we have a molecule that has 2 distinct parts. We have the polar end, or the hydrophilic end, that will be attracted to water. And then the non-polar part that will not be attracted to water. Okay. So you have a hydrophilic head and a hydrophobic tail. You have 2 parts to this molecule. And what that means for membranes is that these molecules are naturally going to arrange themselves so that the hydrophobic parts are hiding. Right? The hydrophobic parts are going to group together and they're going to hide away from the water as much as possible. And so they're going to put their hydrophilic parts on the outside. So in a cell, so this would be like the inside of a cell, and this is the outside of a cell. The inside of a cell and an outside of a cell are almost always going to be made entirely of water. Most environments have tons and tons of water, inside and outside. So the way that these line up is those hydrophilic heads will face the water, right? They all line up together. Those hydrophilic heads will face the water, and they basically block the water out for these tails that stick into the center. Okay, and all those hydrophobic tails are going to stick into the center and clumped together so that they don't have to touch the water. They put the part that is okay with touching the water on the outside, and they keep their hydrophobic parts in the middle. And this structure is incredibly important to the structure of cells. This is called a phospholipid. I just realized I have it written up there. But this is a phospholipid bilayer. It's called a bilayer because there's 2 layers of those phospholipids. Okay, you have 1 layer over here and 2 layers over there. All right, and these phospholipid bilayers, almost every cell type that exists on this planet, for all the way from bacteria to archaea to algae and funguses to animals to humans, everything, all of their cells have a phospholipid bilayer membrane. And why that's critical, why a phospholipid bilayer is important, we'll talk more about later. But the gist of it is that it protects the inside of a cell from everything in the outside world. Because of that double layer with the hydrophobic part in the middle, it means that not anything can get through. It serves as a little shield. So whatever we want to come in can come in, but whatever we don't want to come in has to stay out. It allows a cell to control what is allowed in and what has to stay out. All right. I just realized I am on Do Not Disturb. Okay. All right, and then our last 2 categories of lipids are not as big of categories, but we are going to touch on them. So our last 2 categories of lipids are sterols and waxes. Now, sterols provide a couple of major functions within cell types. Sterols do 2 things. They provide cell membrane strength. So those phospholipid bilayers on their own are what I like to call floppy. Okay? They don't hold much structure. They're very, very fluid and very, very floppy. What cholesterol does, or certain types of sterols do, is they provide structure. They are kind of like, I guess you could think of like if you were making like a brick wall, right? You have a brick wall and you build up all this stuff and it's a strong structure, but say you didn't grout anything. You just piled a bunch of bricks on top of each other. Well, one good kick and the whole thing is gonna fall over. It's a very fluid, loose structure. Sterols, cholesterol, and other sterols like it are basically the mortar, is that the right word? I'm not a handy person. The mortar or the grout or whatever it is that holds the bricks together. So that when you brick them all together and you add the mortar, you can build a house out of that and it's not gonna fall down. So that is what cholesterol does in humans. So this is membrane support in humans and some animals. Bacteria and archaea and algae and funguses have something similar, but it has a different name, but it falls into the same sterol category. But cholesterol, and I know you guys have heard that word before, and cholesterol gets a really bad rap in a human diet, right? You have to eat low cholesterol and all that stuff. The truth is that cholesterol is incredibly important. If you didn't have cholesterol, your membranes would fall apart. But too much structure can be a bad thing. So too much cholesterol can make your cells too structured and too stiff. Okay, so that's why it's important to keep your cholesterol within a certain range. Some is important. You don't want to cut it out completely. You do need it. It's required. You just don't want too much. And then the other major part that sterols form in a lot of animals and mammals, multicellular organisms, is that they are the component of steroid hormones. Now, does anybody know what a steroid hormone is? Can you name any one for me? estrogen. Those are the two big ones, are estrogen and testosterone, which are key to human reproduction. Okay, without estrogen and testosterone, we wouldn't produce eggs or sperm, and there would be no human reproduction. Also, things like aldosterone is one, but I know you guys don't know what that is. But I don't know why I'm blanking on the word, but adrenaline, I can't think of the official word, but adrenaline is another steroid hormone. Cortisol is loosely a steroid hormone. So there's a lot of things in our body controlled by steroid hormones. Estrogen and testosterone are the big ones, right? Without estrogen and testosterone, humans would not be able to reproduce. And those hormones, the base of those hormones, our body actually makes those from cholesterol. So if you're not eating enough cholesterol, again, your body needs some. If you're not eating enough cholesterol, your body will not be able to make steroid hormones. That's why people who are starving, you know, some people who have mental illnesses like anorexia and things like that, and they're not eating enough, they won't go through menstruation or puberty because their body is not producing enough hormone. They're not eating enough food to produce the hormones that they need. All right. And then our last category are going to be waxes. And waxes, as you may imagine, you know, like candle wax and things like that, those are water repellents. Wax is a great way. You coat something in wax and it's going to repel water. Your body also produces waxes. I know it's weird to think about, but you are waterproof, right? If I dunk you in the ocean, you're not going to suck up all the water like a sponge, right? It rolls off your skin. You go stand out in the rain, your clothes will get soaked, but your body loses all the water. You don't suck it in. So it's weird to think of yourself as being like a waterproof jacket, but you are. And the reason for that is because of the waxes produced in your skin, right? Most of us produce oils on our skin. We spend a lot of time trying to get them off of our skin, but they do provide an important function, important service. They make you waterproof. Right? And then we'll talk a little bit, we're just gonna ignore this one. We'll talk more about that later. I don't know why I have it there. Just ignore it, sorry. But waxes are kind of the last category of our lipids. Okay. So our next category of macromolecules, so now we've covered carbohydrates and lipids. Our next one are proteins. And proteins, it is really hard to overestimate how important proteins are. So proteins are the predominant organic molecule within a cell. So there are carbohydrates, there are lipids, but protein is the most predominant cell type within, or predominant macromolecule within any cell. And most of what a cell is and does is controlled or made up of proteins. We're going to talk more about specific examples as we go through. Again, it's really hard to give. You know, with carbohydrates, I was able to say they provide energy. And then fats, it's a long-term energy storage. And membranes, right? I was able to assign a primary job. The truth of it is that proteins do so many things that it's impossible to assign them a primary job. They are in control of basically everything that a cell does. They're incredibly important. And again, you can't overstate how important they are. But they are the predominant organic molecule, and they control just about everything inside of a cell. The structure of a cell, the behavior of a cell, the unique qualities of each living thing, what makes humans different than horses, and what makes each human different from each other is all controlled by proteins. Even things like the differences in our skin. Melanin is a protein, okay? Whether you produce more melanin or less melanin will determine the color of your skin. Everything that happens inside of a human body and any other cell type is controlled and managed by protein. Now, protein is a polymer. Oh God, I cannot spell today, guys. Polymer. Okay, so a polymer is a protein. This is also called a polypeptide. Again, I know everything has multiple names, but a protein is a polymer, also known as a polypeptide. Okay. Now, the monomer of a protein is called an amino acid. Amino acids will hook together individually to form proteins. Now, there are, how many? I think there's 26 amino acids. I think that's right. 26 essential ones. I don't remember. You don't have to memorize them all. You don't have to memorize their names. I have them up there so you can see them. But there are about 26 different amino acids, and they can be hooked together in chains to form an innumerable different number of individual proteins. Because you can have polypeptides that only have 15 amino acids in them in any single order. Or you can have proteins that have literally thousands of amino acids hooked together in a specific order. So the number of possibilities of a protein chain are, like, you can't even count them. There's too many. Now, one of the most kind of crucial and critical ideas when it comes to proteins and biology, and specifically biochemistry, meaning how our biology is affected by the molecules inside of our body, is this idea that the structure of something is going to determine its function. Okay, so a protein's structure is incredibly important to what it does. Many proteins have a very specific shape, and that shape determines what they do. And this is going to make more sense, and we're going to talk about it more when we get to enzymes and things like that. But a really great example for now is something called, something that you see in a human genetic disease. Now, you can see over on the bottom, this is what a normal red blood cell looks like. And this normal red blood cell has a protein inside of it called hemoglobin. Hemoglobin is a protein. And this protein has a very, very specific shape. I always think it looks like a little inner tube, right? That is why red blood cells have the shape that they do. They form a little cup. I think they look like a little inner tube. I've heard like donut, or somebody says checker piece, like the inside of a checker piece. But they form this little cup you can see this picture here. It's wider at the edges, and then there's like a divot in the middle. Okay, right here, this is like a little cup. And the purpose of hemoglobin carries oxygen through your blood. And literally, the shape of this hemoglobin is critical to it being able to do that. I like to think of it as an inner tube because hemoglobin, or oxygen, will literally sit in that little cup of hemoglobin. It sits right in the middle, and it rides the inner tube down the lazy river that is your bloodstream to get it where it needs to go. Okay, that shape is exactly where the oxygen goes. And that shape determines its function. Now, something can happen in a disease called sickle cell anemia. So sickle cell anemia is a relatively common illness. It's not super common. Does anybody here have it or know somebody that does? It's usually, you have one person over there. So it's usually about a 50-50 shot if a couple people raise their hand. Sickle cell anemia is not uncommon. It's relatively common. And what sickle cell anemia is sickle cell anemia is a genetic disease where that hemoglobin, that's not how you spell that. Hemoglobin. That's the abbreviation for hemoglobin. But hemoglobin is altered in shape. So sickle cell anemia is the result of a genetic mutation that changes the shape of the hemoglobin protein. Okay? And instead of being this nice little rounded cup shape, the hemoglobin changes to what is called a sickle shape. And the sickle shape is kind of like a crescent moon or a canoe, however you want to look at it. But this hemoglobin does not have the nice little inner tube cup shape. And these sickle cells are incapable of carrying oxygen. Okay. So these sickled red blood cells, because of one change to that protein, the entire shape of that protein changes, and it is completely non-functional. It is no longer able to do its job because it is not the correct shape. So it's going to be a common theme when we're talking about proteins that the structure or the shape of the protein is going to be critical to its function. And with sickle cell anemia in particular, sickle cell anemia is a devastating illness. It's a long-term illness. We've gotten to a point in healthcare now where people who have sickle cell anemia can live full, relatively normal lives. However, that life is filled with pain. It's an incredibly painful disease because these red blood cells with their pointy ends don't flow through our blood vessels the way that they're supposed to. So those red blood cells get stuck in joints and in organs. They can cause organ failure. It's incredibly painful. Most people who have sickle cell anemia have to get transfusions every couple of weeks so that they can carry enough oxygen, so they have enough normal blood cells to carry enough oxygen to keep them alive. So sickle cell anemia is a survivable illness, but it's a very difficult one. It's painful, and it involves a lot of trips to the hospital. When I used to work at a small hospital, we had like 3 people that came in literally every week to get their transfusions. So with sickle cell anemia in particular, sickle cell anemia, that change to the shape is the result of one single amino acid change. We'll talk more about this later. But in the hemoglobin chain, one amino acid is wrong. So when that protein was being built in the genetics, one single amino acid is wrong. One single amino acid being incorrect in one single protein within a human body causes them to have a devastating illness that will last their entire lives. One amino acid. And so it's a very delicate process. Proteins are incredibly important, and their structure is always going to affect how they act. Okay. So protein structure, speaking of protein structure, because it is so important, protein structure is broken down into 4 pieces. We have 4 layers, I guess, of protein structure. The primary structure of a protein is just the order of the amino acids. In the chain. So remember, a protein is just a chain of amino acids, and they can be in any order, and that order is determined by a lot of things that we're not going to talk about yet. But that order is incredibly important to the final structure. So the first layer of building a protein is the order of the amino acids. Okay. Now the secondary structure, there are 2 types of secondary structure. And again, we're trying to keep this as surface level as possible, so don't get too hung up on the details of this. But there are 2 types of secondary structure of a protein. We have an alpha helix and a beta pleated sheet. A helix is like a corkscrew, right? You can kind of see it here. It's like a corkscrew. A pleated sheet is more like that. Okay? But the secondary structure of a protein is mostly based on how amino acids react with the amino acids next to them in the chain. Okay, so the primary structure again is just the order of the amino acids. The secondary structure is how those amino acids react to each other dependent on the order. So we put them in a specific order, and it's how each amino acid interacts with its neighbors, the close by amino acids. And they will either form, depending on the order and where they are, they will either form an alpha helix or a beta pleated sheet, depending on the amino acid and how it interacts with its neighbors. Now, the tertiary structure is how those alpha helices and beta sheets interact with each other. So we have this one amino acid chain, right? And the local folding, so you'll have this one big chain and say that the first bit is an alpha helix, and then the next one will be a beta pleated sheet, and then the next one is another alpha helix. And then maybe we have an alpha helix for a while, and then this will be a beta pleated sheet. And then what will happen is these alpha helices and the beta pleated sheets will interact with each other. And the protein basically folds up into this three-dimensional structure based on how those things interact with each other. So in this one, you can see this is an alpha helix, this is a beta pleated sheet, and they fold it up to form this 3D complicated structure. In this one, it just looks like a big tangle, but this is the shape of the protein. It's the three-dimensional shape. Okay. And then the quaternary structure, or the fourth layer of structure, is how multiple chains interact. Okay. And so not every protein has quaternary structure, but sometimes individual chains will come together to form an even bigger three-dimensional structure. So the quaternary structure is how those individual chains, or how those multiple chains, interact with each other. But what this means overall is that every single protein will have a unique shape and a unique structure that will only interact with the molecules that it needs to based on that specific shape. And again, if you mess up the shape of a protein, you mess up the function. Many proteins, if you alter their shape, they no longer work. And that is the basis of a lot of genetic illnesses. Things like cystic fibrosis and Tay-Sachs disease and sickle cell anemia and Huntington's disease. All kinds of things. A lot of genetic diseases that you've heard the words for, ALS, stuff like that, those are all based on protein mutations and having the shape of a protein change from what it's supposed to be. Okay. All right, so our next one is nucleic acids, and this is our last macromolecule. And remember that this is our polymer up here, and then our monomer are nucleotides. So nucleic acid is the polymer, nucleotides are the monomers. And there's two major types of nucleic acids. We have DNA and RNA, which I think are things that most of you have heard of in your life at some point. DNA is mostly used for, and again, we're gonna get more into DNA and RNA in a later chapter. But DNA, for now, its major function is hereditary information storage and reproduction. It's how our body stores genetic information, and it is how we transmit genetic information to the next generation. So how any organism is able to reproduce. right? Humans reproduce sexually, so do most animals. Bacteria don't reproduce sexually, but they do reproduce. Okay, they have to pass their DNA on to another generation, and that is done through DNA. RNA is mostly responsible for producing proteins. And we're going to get more into the details of that later, but RNA's major purpose is what is used to make those proteins. All right, and like I said before, the monomer of our nucleic acids are nucleotides. Okay. Now, a nucleotide is going to be made up of a couple of different things. The first thing it has is a phosphate group. And again, you don't have to memorize these individual structures, but you do have to be able to name them. So you have to be able to say that a nucleotide has a phosphate group, a sugar, and a nitrogenous base, right? I'm never going to ask you to draw a nitrogenous base. I'm never going to show you a picture of a nitrogenous base and ask you to tell me what it is. But you have to know the three components of a nucleotide. So a nucleotide is made up of a phosphate group, a pentose sugar, which means a 5-carbon sugar, okay, pentose means 5-carbon, penta, like the pentagon, has 5 sides. Pentose sugar, a 5-carbon sugar, and then a nitrogenous base. Nitrogenous means that it contains nitrogen. And there are 5 different nitrogenous bases. And again, you don't have to memorize the structures, but you will have to know the names. So the nitrogenous bases are adenine, guanine, thymine, cytosine, and uracil. And we usually refer to these by their first letter, A, G, T, C, and U. Hopefully some of this is coming back. I know all of you took some high school biology at some point. Probably talked about this. Okay? And the way that DNA and RNA are formed is these individual nitrogenous bases are linked together, again, in a big long chain. The monomer is a nucleotide, and they will hook them all together in a big long chain that then becomes nucleic acid, either DNA or RNA. Okay. So this is kind of a complicated picture, and we're going to cover a lot more of this later. So all you have to know for now is the basic structure of the 2 different types of nucleic acids. Over here we have RNA, and remember again that RNA is mostly responsible for protein synthesis, and DNA is responsible for reproduction and information storage. So the basic kind of structure of nucleic acids, the 2 different types, DNA is double-stranded. It forms a double helix. And again, hopefully this is some word that you've heard before. Most people have heard of a double helix. But what that means is what you can see over here is there's basically 2 chains of nucleic acids. So you have 1 chain of amino acids right here and 1 chain right here. And those 2 chains interact with each other to form a double-stranded molecule. Okay, so we have 2 individual strands, and they interact with each other and form this big double-stranded twisty helix. Some way people think about it is like taking a ladder with the 2, ends of the ladder, the legs of the ladder, being the 2 strands of DNA, and then the middle is where they're interacting with each other, and then you twist it up. So you took a ladder and twisted it. Okay, so double-stranded. And the nucleotides in DNA, and again, we're going to go over this in more detail later, but DNA uses C, G, A, and T. So cytosine, guanine, adenine, and thymine. Of the 5 of the 5 nucleotides available, DNA only uses these 4, C, G, AT. RNA, on the other hand, is single-stranded. As you can see, there's only one strand of nucleotide bases. You can see kind of the more molecular picture down here. There's only one strand of nucleotide bases. It does form a single helix. It does still form kind of a twisty molecule, but it is single-stranded, not double. DNA is double. DNA is double. Okay? And RNA also only uses 4 of those nucleotide bases, but it uses C, G, A, and U. So the big difference here is this one at the bottom. In DNA, you have thymine, and in RNA, you have uracil. The other 3 are in both. So cytosine, guanine, and adenine are in both RNA and DNA, but in RNA, you have uracil, in DNA, you have thymine. All right, and then the last thing I'm going to cover about nucleic acid is something called adenosine triphosphate. And the reason I'm talking about this here is adenosine triphosphate is a molecule that uses that adenine, that same adenine that is in DNA and RNA, but it uses it as something else. So it's called adenosine triphosphate, and it has an adenine group, that same sugar, but instead of a phosphate group, a single phosphate group, it has 3. It has 3 phosphate groups. Now, adenosine triphosphate is also known as ATP, which again may or may not be something that you have heard of before. ATP is the energy molecule of most cells. ATP is how every type of cell stores, creates, and releases energy. Okay? That's how a cell powers everything that it does, is through the creation and breakdown of ATP. Right? And we'll talk more about how ATP works later, but I bring it up here because that structure is very, very similar to that nitrogenous base, or that nucleotide base. Right. All right, so those are the major kind of components of cells, the 4 big macromolecules. And like I said, we're going to spend a lot of time later on talking about each of those more individually. The second, as a fair warning, I guess, the second test in this class has a lot to do with the structure of proteins, how DNA is made, how RNA is made. A lot of people think it's the hardest test as exam number 2 because it does get fairly technical. Okay. But now we're going to move on. Now that we've talked about those basic components, we're going to talk about the things that make a cell a cell. So what is it about a cell that makes it a cell? What does something have to have in order to be considered a cell? Now, a cell, if you don't know, is the smallest unit of life. So in order to be considered alive, a cell or an organism can be made up of cells. Now, an organism can be made up of one cell. One single cell can be an organism. And that's why we call it the smallest unit of life. So you can have what's called a unicellular organism, which is an organism, again, made of one cell. Things like bacteria and algae and archaea, even some types of yeasts and funguses, those are unicellular. They are made up of one single cell. The whole organism is one cell big. Or you can have a multicellular organism. mean that it's made of many cells, or at least more than one. Okay, so humans and animals, a lot of molds, worms, insects, plants, those are all made of multiple cells. They're multicellular organisms. But again, something to be considered alive, something has to be made up of at least one cell. And all cells share a few physical characteristics. There are a few things that have to be true in order for something to be considered a cell. All right, so there are 4 characteristics that a cell has to have in order to be considered a cell. The first one, or the first 3, I guess, are these ones right in the middle that these 2 different types of cells share. So the first one is a cytoplasm. And the cytoplasm of a cell is the gel-like substance on the inside of a cell. Okay, so it's every single cell on the inside of the cell has this kind of jelly-like substance that basically everything else floats around in. So all of the other components are going to be sitting in the cytoplasm. It's just what is on the inside of a cell. The second thing that all cells will have is a cell membrane. We talked about that a little bit before, but the cell membrane is the boundary of a cell. It's what separates the inside of the cell from the outside. And the cytoplasm is all contained on the inside of that membrane. So on the inside of the membrane is all the cytoplasm and everything else that that cell is made up of. The third thing that all cells have is ribosomes. And ribosomes they're made up of a couple of different things, but ribosomes are structures used to make proteins. And like we said before, proteins are the central kind of lifeblood to any type of cell being able to do anything. A cell doesn't exist if they don't have proteins. They have to have them. And ribosomes are the things that make proteins. So in order to be considered alive, in order to be considered a cell, you have to have a way of synthesizing or making your own proteins. And that is what ribosomes do. So all cells will have some type of ribosome. And then the fourth thing that all cells will have is genetic material. DNA, genetic material. So to be considered alive, you have to be able to reproduce yourself. It is the basic function of most organisms, whether we like to admit it or not. One of our main purposes in being alive, one of our main instinctual drives is to make more of ourselves. So reproduction is a central part of every organism, every cell type. And in order to do that, they have to have DNA or genetic material. So those are the four things that every single cell has in common. Now, we are going to spend a significant amount of time, I'm going to kind of skip over it for now, but I am going to spend a significant amount of time later talking about our two major categories of cells. We talked about this briefly, but we have prokaryotic cells, which is bacteria and archaea. Go back a couple of slides if you don't remember that. But our two major cell types are prokaryotes, like bacteria and archaea, and then eukaryotes. Remember that you are a eukaryote. So our cells are eukaryotic cells. Animals, plants, funguses, Those are all eukaryotes. And we're going to talk about the differences between them in more detail later. However, the big difference between a prokaryote and a eukaryote, there are many differences, but the biggest one, and the one I'm going to talk about the most, and this is going to haunt your nightmares, I'm going to say it so many times, is that a prokaryote does not have a nucleus. Okay, so I'm saying it now so that we can remember it. And a nucleus is a separate little holding spot for genetic material, basically. But in prokaryotes, they do not have a nucleus. Their genetic material, their DNA, floats around in their cytoplasm. They do not have a special little room for their DNA to sit. Eukaryotic cells have a true nucleus. Okay. Like I said before, there's many differences, and we'll talk more about some of the other ones later, but this is the big difference. Eukaryotes have a nucleus, prokaryotes don't. Always. I think I've said this before, that there are very few things in biology that are always or never. Okay, this is one of those times when it is always. Prokaryotes always lack a nucleus. Eukaryotes always have one. Okay. And then we're just going to talk a little bit about some kind of definitions and nomenclature. We're going to talk about taxonomy and identification. So taxonomy is the science of identifying living and classifying living beings. And so taxonomy, one of the things I always like to say is that humans, for good and for bad, you know, this isn't always a good thing, sometimes it is, but one of our favorite things to do as humans is we like to understand stuff. And the easiest way for us to understand things is to put them in boxes, right? We like to have a nice labeled box for everything. We want everything to fit in an exact thing. We want to be able to put something in a box and slap a label on the outside that says, this is what goes in this box. Okay, it makes us feel calm. We like it. It helps us understand things, and a lot of times that's a really good thing. Sometimes it's not, but science, a taxonomy is the science of doing that. It is a science of taking all living beings and trying to put labels on them so that they fit in boxes, so that we can understand how they are similar to each other and how they are different to each other. And taxonomy typically uses 8 levels of classification. And it goes from least specific to most specific. So up here at the top is the least specific. And that is the domains. Now, we already talked about the 3 domains. You can go back a couple of slides if you don't remember. But the 3 domains are bacteria, archaea, and eukarya. Again, go back and look at the tree of life if you don't remember. But those are the 3 domains. And what I mean by least specific is that those boxes, right, those are big boxes because every organism on the planet fits into one of those 3 boxes. It's either a bacteria, an archaea, or a eukarya. So each of those boxes is going to have hundreds of thousands of organisms within each box. Okay, that is the least specific. And then at the bottom, we're going to only really worry about domain, and then we're going to talk about genus and species. So those are the only 3 that you have to remember, is that domain is the least specific, and then down at the bottom are most specific is species. So in an ideal world, when we get down to the species level of identification, there is only going to be 1 organism in that box. Okay? And so the science of taxonomy is basically taking smaller and smaller and smaller boxes and labeling them so that we can fit everything into their correct box. Okay? So this is kind of an example of how we classify something Again, classification is the process of putting those organisms into these labeled boxes. Classification is done in all eight levels, starting with the most generic. So in this example here, we're looking at two different organisms, but we can kind of look at each level of classification. So at the top, we have domain eukarya. So domain eukarya is an enormous box that contains all eukaryotic organisms from molds and yeasts all the way up to humans. And that's a lot of organisms. Our next layer of classification is kingdom. And you don't have to memorize these in order. Like I said, you have to remember domain at the top, species at the bottom. But next we have the kingdom. And we can separate these two kingdoms. There are many other kingdoms, but we're only looking at two here. So kingdom Animalia. Kingdom Animalia, the class or the requirements to be in kingdom Animalia are to be eukaryotic, heterotrophic, and usually multicellular. I know you guys don't know what those words mean. That's fine. You don't have to. But what you have to see here is that we are using more and more specific characteristics to create smaller and smaller and smaller boxes. So all of those organisms at the top, our giant box of eukarya, are then going to be broken up into smaller and smaller boxes. And hopefully by the time we get all the way down to the bottom, we are going to have a single species sitting in a single box. For example, this one down at the bottom is genus Homo, species sapiens, which are humans. And those are the characteristics right here that make Homo sapien separated from its nearby relatives. Okay. And then over here we have genus Paramecium, species Caudatum. So that's a paramecium. It is also a eukarya, so that's something to, both of these organisms are in the domain eukarya. Paramecium is a parasite that lives in the water. It's a single-celled parasite. Okay, now phylogenetic trees, phylogenetic relationships are a little bit different. The difference between taxonomy and classification and phylogeny is in phylogenies we are looking at how those boxes are compared to each other. So instead of just looking at something in its box and saying this has this characteristics, we're saying, okay, but this box has this and this box has this. How do they relate to each other? How closely are they the same? How far apart are they? How closely related are they? And that is what phylogenies do. Phylogenies take relationships between species and genuses and kingdoms and all of those other things into account. Okay, so these take relationships between organisms into account. Instead of looking at these organisms, Instead of looking at these organisms as individual boxes and trying to separate them out as much as possible, we are looking at them and how they compare to each other. And this is the most updated tree of life or phylogenetic tree. And this shows how organisms in these different kingdoms relate to each other. And this is actually a 2-scale thing. And the distance between the organisms on the trees is related to how closely they are to each other. So an organism like an entamoeba and a slime mold are actually really relatively close to each other. They're very close. Their lines are physically close to each other on this tree. So they are relatively close to each other. However, an Aquifex bacteria and an animal are incredibly far apart from each other. So they are not related to each other very much. Okay, so that distance matters. Okay, and so this is the most commonly used method of talking about organisms and how they relate to each other as of now. But there's actually a lot of data that shows that this tree of life is even outdated. The tree of life is constantly being updated. But there are a lot of thoughts now that this tree of life is outdated, and the more accurate picture would be a web, because the way that bacteria and viruses transmit information back and forth and cause mutations, all of these organisms are actually more closely related to each other than a tree would suggest. They're actually interacting in a lot of ways. But as of now, this is the most accurate representation of how organisms interact with each other. Okay? All right, I have 1 more minute. We're going to cover this. Don't pack up until I'm done, because once everybody starts packing up, it gets too loud. We can't talk about it. All right, so when we are talking about bacteria and organisms, we are going to talk about them in what is called binomial nomenclature. Binomial meaning 2 names. And we are going to talk about them in their genus and species. So we will spend a lot of time talking about the genus and species of a particular organism. And when you are writing, this is less important in this class, but if you take lab, this is incredibly important. Organisms are always written in italics or underlined. If you are writing it by hand, you can underline it. The genus of the organism is always capitalized, and the species is never capitalized and never shortened. So when we're talking about this organism here, this organism is domain bacteria, order Enterobacterialis, family Enterobacteriaceae. You guys don't care about that. But the genus is Escherichia, and the species is coli. Its binomial full name that we refer to it as commonly is Escherichia coli. Notice that the E in the genus, Escherichia, is always capitalized. The coli, the species, is not. However, you can abbreviate Escherichia coli into maybe something that more people recognize is E. coli. But you can never shorten the species name. So something really common, there's a bacteria that's called Staphylococcus epidermidis. That's a very long name, Staphylococcus epidermidis. You can call it S. epidermidis. You can shorten the species to S, but you cannot call it Staph epi or S. epi. You cannot shorten the species. Okay? All right, thank you for waiting. You can go now.